So with the title of biomolecules, it should be abundantly clear we're going to go back down to the molecular level, but keep in mind that the word bio means life. In this case, we're talking about biology. So any molecules that have to do with life, we've actually been able to categorize into a particular group called biomolecules. So the learning goal for this whole section, this topic, is to differentiate between four biomolecules based on their structure and function to support cells. Before we get into the four, there are six elements you should know. These are hydrogen, carbon, oxygen. Now, it is very neatly highlighted for you that hydrogen, carbon, and oxygen are by far the most important ones, but just as important, or maybe not just as important, I just literally said they were more important. Nitrogen, sulfur, and phosphorus are less important. However, uh, they are still required in most life. Like you would not find life without a lot of those three. So the top three for sure: hydrogen, carbon, and oxygen. I've often heard of them shorten it to CHO, so that you can remember it as those are the three, or just CHON because nitrogen is pretty high up there as well. But if you really want to, the whole group together could be CHOMPS with an N. So, as a quick preview, we are going to talk about these four: carbohydrates, lipids, nucleic acids, and proteins. So, there is a concept in chemistry. This is a huge preview. Those of you actually planning on going to med school, going into further into biochem, or just molecular and cellular biology, or just biology in general, you will probably need to know the term functional groups. And these functional groups are common patterns. As you learn how molecules come together, you'll see that they're actually very natural patterns that almost must appear. But these are the some of the most important ones. Now, we're not going to make you guys memorize these in the future in some of the more advanced classes. I believe AP Chem does, but for sure, organic chemistry in college will definitely ask you to memorize them. You will have things like aldehydes, carbonyls, carboxylic acids, esters, ethers, and these are all things that you're going to want to memorize. If you don't start now, definitely by the time you're taking the MCATs, because I think even though they give you a handful, it is just useful to have them. It's like multiplication table. If somebody handed you a multiplication table, you could go and find the answer, but it's just that much faster to solve a problem when you have these memorized. So, what you want to think about these functional groups is that they're combinations of those six elements. If you investigate any of these, you'll notice that um, all of them are mostly CHO or CHON. Very rarely will you see a P or an S in there. But you'll also notice that there's R. You can ignore the R for now. We can talk about that later. But essentially, it just represents a whole chunk of another molecule. So if we're talking about amino acids, we just like meh, just ignore that for now, okay? But these are functional groups, meaning that these are um, the most important elements that help you identify. This is very interesting because we'll talk about this in more detail. But for now, just recognize that there are some very common combinations using CHON and CHOMPS to figure out, or not to figure out, but for organic materials to usually get together. Okay, so now we go into some vocabulary that categorize biomolecules. The first one is a monomer. So mono means one, and so you'll have a single unit. We'll talk about those in more detail. But for now, we just mean that there's one piece of something. The next one is polymer. So monomer and polymers. Polymers are many monomers. In the case of this yellow circle, they're going to be chemically bonded. Now, this is very important, okay? You can't say there's just a pile of them. You have to make sure that you're saying they're chemically bonded to each other to build polymers. Without that, it doesn't work. You'll notice in the top right corner, we're saying that's not a polymer. It has to be repeats of the same thing over and over and over again. If you start combining random other things, we have another name for those, macromolecules or whatever else. But if we say polymer, it means it has repeated units that keep building on top of each other. Okay, so there's actually quite a bit of chemistry that goes behind how polymers are formed or broken. We will introduce, this is definitely not middle school level, so we're only going to go like, we're going to dip our toes in just a little bit, okay? Just put your right foot in and then shake it all about. The first thing we're going to learn is condensation. Condensation, if you remember what you've hopefully heard by now in normal day life, is usually some kind of droplets of water showing up because it's either too cold or too hot. It depends on which perspective, but generally speaking, there's some water coming out. This is one way to help remember what condensation is. So, a condensation is water is removed. So suddenly you start getting water showing up when this reaction happens. And what happens is that you create a bond as a result. If you see over here, there's an H and there's an OH. When they combine, those become H2O, thus creating a bond between the two. Now, there is a specific term of dehydration synthesis. You can ignore that for now, but just so you're familiar, this term will come up a lot. It is a much more specific version of condensation. For now, just memorize condensation. Water gets created, and you leave behind two hand-holding molecules now that create a much larger molecule. So when we talk about polymers, there are two monomers that will join, and there's usually water that gets created. Okay, keep that in mind. It's actually a very important bit to remember if we do FRQs. One of the clues is that, hey, a lot of water gets formed as a side product. So you should use that to help clue you in going, oh, something's forming, something, a lot of it's being created. Okay. The opposite of that is called hydrolysis. I don't have any neat ways to memorize this, so hopefully Mr. Vo or even myself will come up with a way for you guys in class. Worst case scenario, you come up with your own or looks online. Somebody will have some neat way to remember. I don't know why. It's not what the word means at all, but to me, lysis makes sense as a cut. So this diagram down here, I should probably cover it up. It's not the greatest example because it's actually the opposite. This is actually another example of condensation up here. So this diagram down here, if any of you are getting confused, don't worry. It's just we accidentally put another one as an example. It would be the opposite. If you cut maltose and it became glucose, you had to insert the opposite. They're basically opposite of each other is one of the points we're trying to make here, and is actually immediately back and forth. And so hydrolysis, the covalent bond is broken, whereas condensation, covalent bond is formed. So in hydrolysis, you add water. Now, you can't just literally pour water on some stuff like this and break it. There are usually other things involved, and we'll talk about what those are when we talk about actual chemical reactions. For now, just remember that in order for this to happen, you have to be able to insert the water back in so they can form back into those original bonds. So it's condensation to add, hydrolysis to break. Again, I don't know why lysis makes sense to me as cutting. I think I did enough chemistry after a while. You hear this lysis, that lysis, and you're like, ah, it's about cutting, it's about breaking a bond somewhere. So to me, it makes sense. So to illustrate, that's what happens. So here's a neat chart some people come up with. This is an excellent way if in case you ever come across two major concepts, two major definitions, or vocab words to really compare the two with each other. You'll see that they, for the most part, are opposites. So we talk about hydrolysis over here. The definition is that the breakdown of a polymer into monomers upon adding water. But on dehydration, it's removal of water instead of adding water, and now you bind the monomers together to form a polymer. Again, hydrolysis breaking the polymer into monomers. Here, you're binding monomers into a polymer. The origin, to help you, hydrolysis. Lysis means breakdown, and hydro means water. Dehydration, D means removal of, and hydro is for water. Water molecules are added, or water molecules are removed. Types of reactions. So cold decomposition and synthesis, you won't know what these are until you've taken some basic chemistry. If you're lucky enough, and people like your Mr. Robson or Miss Wilkie cover this, you'll understand what we mean by this. But in the meantime, just know that it's essentially an even larger category of what we say when we say breakdown. Anytime there's something big and it breaks to pieces, we call it decomposition. Synthesis, when you have many small pieces added up, it becomes one whole piece, right? So the type of products, monomers, and the types of products for dehydration are polymers. For byproducts, generally there's no byproducts in the process of hydrolysis, but for dehydration, it's water molecules. Arguably, if you're gonna say byproducts, keep in mind, strictly speaking, and this is purely academic, byproducts means you did not intend for them or they're not worth your consideration because that's not why you're doing the reaction. So byproducts usually means something that shows up and you're like, get out of the way. In the case of dehydration, you don't really care for the water, like that's not at all what you're looking for. So we call it a byproduct as it leaves. However, when we talk about actual products, there is hydrolysis over here. You're saying it's the monomers, right? If you're talking more colloquially, some people might say monomers are the byproducts. Please do not say that. That is not okay to say when you're in a science classroom because now we have to use academic language. We have to be very clear what we mean. There is a difference between products and byproducts. Again, it's all about intent. Did they come out of the reaction? Yes. If we wrote a balancing equation reaction, you would see it written on the right side. However, again, byproducts we're not interested in. They're not why we did this reaction to begin with. So that's why we say there's no byproducts because nothing extra comes out. Next, we have examples, digestion of proteins, lipids, and carbohydrates. A lot of our body, when we do this to create energy for ourselves, this is hydrolysis. Keep that in mind. This is useful for remembering when you're talking about FRQs, okay? For dehydration, we have alcohols to form acetyl and ethers. There's a star here. I'm not sure where Mr. Vo's getting at. But if I find out, I'll let you know in class, and I'll make sure to add an addendum. All right, so now we get into the meat of it. This is the main section of this topic. We're going to start with carbohydrates, sugars. Personally, my favorite, anything made of bread, anything that's got sweets in it. Plenty to talk about here. First of all, understand that Mr. Vo wants you guys to memorize the name of the monomers and the polymers. So for each one, you're going to want to memorize and remember there's one of each. So for now, my recommendation is just to remember, well, what are the names? What's a monomer? What's a polymer? So at least one card, one Quizlet Ants question is dedicated only to, well, what are their names? The monomer of a carbohydrate is monosaccharide, okay? It's spelled, this is just old Greek or Latin. I always can't remember. I never took Greek or Latin, so I don't know the differentiation. They're all the same to me personally. So it's saccharide, Sacharide. Now, if you guys horribly misspell this on the test, I will have no choice but to take points off. As long as you're close enough, it's okay. But it is a weird spelling. We're not used to seeing double C's followed by an H. So anyway, it's mono. I used to pronounce it saccharide just to help me. If that helps you too, then it's monosaccharide. But these are simple carbohydrates, okay? Like glucose, fructose, galactose. Don't get used to this. There are naming conventions that lasted and survived for a long time. People started making names for molecules for hundreds of years. We have now a new version where we try to name everything without using some of the older systems. But there are some that bled through. So you have glucose, fructose, and galactose. Anything that ends with O's, very generally speaking, is a sugar. It's a carbohydrate, okay? Now, the polymer is known as a polysaccharide. Again, polysaccharide. In this case, I think they're very easy. You just remember that they're saccharides. Literally, saccharide, I think, just means sugar. I have to look this up. Mr. Vo probably knows. But you have mono and poly. That's it. So monosaccharide, polysaccharide. Now, these are complex carbs. And to help you remember this, there are things like cellulose, lactose, glycogen, and starch. One way to remember this is that there's a fun experiment. I don't actually have the tools right now. It's not an experiment, really. It's just a demonstration. There are two ways. You can either take sourdough or any bread, for frankly speaking, because they're made of starch, and put it in your mouth. You have in your saliva a specific, what's it called now? A specific enzyme that breaks down the starch molecules into its individual sugar. If you put sourdough into some water and then you test for any sugars, it'll come out negative. But if you start spitting in it, let your enzymes get to work, after you wait a little while, it shouldn't wait more like 10 minutes, you'll start actually detecting sugar, the presence of sugar. And that's because you're breaking them back down into monomers, right? So here's a great example of I've used this now, so it probably won't show up on the actual exams, but something like this where I say, hey, we had a bunch of starch, we broke it down to pieces. Your brain should immediately go to, oh, how do I break it down? Was it dehydration slash condensation, or was it hydrolysis? Try to remember that starting now. If you're still like, oh, I'm not sure, you need to start memorizing immediately. Okay. And for those of you who are like, hey, I want to learn how to one shot just listening to a lecture and not have to memorize anything or write it down, this is how. As I'm talking, you're already like, aha, you are talking monomers and polymers. So A, I have to memorize this. B, are there any clues to help me learn about these things? Like giving you a clue that anything ends with O's. This may or may not help you because I'm not sure that we're going to test you on what O's means. That's definitely not a testable thing, at least in my mind. But for sure, you'll want to know that you have these things being broken down back into monosaccharides, or if you build them back up, you're turning into things like starch or glycogen, right? Now, the bond itself is called a glycolytic linkage. And that's the specific O that's right there. You'll see it. There's always an O right between those sugars. That is what's known as the glycolytic linkage. Mr. Vo's way of memorizing this is when you say sack of potatoes and sack of sugar. A lot of times they're still sold that way. Sugar is still either in a bag or flour, so sack of makes a lot of sense. So you say mono or poly saccharide. Get it? So Mr. Vo has it so that it says the primary function is short-term energy boost. Now, this is not wrong, but it's a very strict perspective of, say, or like large organisms like humans and other mammals or reptiles or whatever. If they happen to feast on something that has a lot of carbs, they will get a short-term energy boost. In anatomy, you'll learn a lot more about why. For our class, I don't think we'll go into that much detail. If we do, I'd be surprised and I'll be very happy. I'd love to talk about it, but for the most part, there's nothing to really talk about here other than it's an energy boost. However, I will make a contention. For our tests, I will be writing it down as short-term energy boost, but I want you to keep in mind that in general, because one of the main things that carbohydrates is known for is glucose, glucose is an energy source. It is not the only energy source, but it is depended upon by so many large multicellular organisms, including plants, that I would argue that the primary function of carbohydrates is the main energy source. Rather than the short term energy, it's the main one. It's the one you want to usually work on. When we get to lipids, I'll explain why I consider it the main one and why I think most in the scientific circles would say that it is the main. But for the intents and purposes of our class, think of it as short term. And the reason is because we're going to talk about lipids. Now, the secondary, and this is where it gets a little bit tough for a lot of people because you're thinking like the picture on the side, a lot of candies, rice, grains, and you're thinking, oh, it's energy, it's going to make me feel great. But now we get to secondary, and this is where you don't think of sugars this way. But once you study this a lot, it becomes second nature. So we're going to ask you to start memorizing starting now. Sugar is not just for energy, there's a lot of reasons to have them. One of them is to build cell wall and DNA. It's actually a major part of their structure. And you guys will see that when you get into the molecular structure of them. The communication is another secondary function. The surface receptors of blood types are, I would say, baseline sugar. It's not 100% sugar, like the same way most things are, but it's pretty close. This is why, and again, we'll talk about this in a lot more detail. There's no need to right now. Just remember, you're going to have to memorize this. When I say what are carbs for, the first thing and most important one by far and large is that it's for energy, right? The second one is structural and communication. So the second of our biomolecules is lipids. You have the monomers, and the polymers again. This time the monomers are a combo. You're going to have to memorize this is a little bit different than the rest. It's a glycerol head. That's why it's circled by itself over here. That's glycerol. And then fatty acid chains. Now the R you don't have to worry about. It just means there's a lot of chains. In fact, if you want, it can be previewed down here. Those, uh, up and down, up and down, up and little waves. Those are going to be what the R's stand for, and those are the chains of fat. So for now, don't worry about it. Just know that if I say what's a monomer, you're like, well, it's got a glycerol head and it's got 3 fatty acids, okay? And that's usually what we're going to consider. Well, not 3 fatty acids, just a single fatty acid usually, and that's considered the monomer. Now, example of these are things like oil and butter. You guys won't need to worry about this because you don't need to memorize it. It's just an example of what they are. Now, polymers are the triglycerol or triglycerides, as some places are known. They're different things, but anyway, the examples are triglycerols, phospholipids. If you remember this in cell membranes, I don't know if you guys have covered this yet, but the idea is that these guys are just one part of millions of them making up a single cell membrane. And that's what the polymer looks like versus the monomers down here. The bond is called an ester linkage. Don't forget, we're going to be memorizing 3 of these for each one. So there's monomers, polymers, and then the bond itself. So this one's called an ester, and it looks like what's highlighted there. The CO, then another CO. But this specific bit is called an ester linkage. So fats and lipids, their primary function is energy storage, much more long term. This is why Mr. Vo had it compared to carbs. You think short term, and then lipids are sort of long term. In diets, especially those of you who play sports, you should know anything that has a lot of fats in it, any kind of oils, it'll take much longer to digest. And so you really don't want to, because a lot of times it will just drain a lot of your energy to try to digest. So you can't have that burst of speed. Right before a race, especially if it's only an hour away, you definitely want to eat something carby. That's why there's a lot of times there's just like that energy bar. They call it an energy bar. It's just like bunched up of carbs and whatever else stuffed in with a little bit of some other things so you don't just completely crash after a quick burn. But the people have been studying this stuff for a while, and we've known this for a long time. People knew it, like, oh, you really want a long-term food? You make sure to have something fatty in there. As with carbohydrates, there are secondary functions. The first one is also structural. You need to build things like cell membranes. Instead of cell wall, we're talking about cell membranes here. This one can be easily confused. Would warn all of you who are memorizing this part here that cell membranes are made up of, as you can see these diagrams, those pink circles are the glycerol heads, and the little tails are the fatty acid chains. So that's what we mean by they're made up of fat lipids. I can't even talk right now. So your smooth ER basically blobs out little bits of these guys, and when they fuse into the cell membrane, they become part of it. Now, they also make vitamins like vitamin A, E, D, and K specifically. The other function then is communication. They become things like hormones and steroids. You will learn later when we talk much more about cell membrane and cell membrane transport why it's important for these hormones to be made up of lipids. For now, just remember that when we have hormones, which are the main chemical signals of your body, it needs to be made up of lipids in order to communicate. The last bit is insulation. This helps retain heat on a larger, much larger multicellular level. Um, a lot of animals really depend on the fat to survive. You'll see animals like polar bears, anything that hibernates, especially in the Arctic oceans, anything that dives deep into the ocean, they're usually coated in a gigantic, thick layer of fat. It is well known that when you hunt animals like walruses or whales, they have a specific type of fat known as blubber, and it is extremely good. Like really good at insulating and keeping the heat that their body creates against the cold water. Most of us, if we ever jumped into water that cold, would not survive longer than, a few hours. But those animals can live their whole lives in those icy cold waters and be fine. For this section, it is great for you guys to take some practice just to see how well you guys can keep up with it. I'll reveal the answers, so I'm going to ask you to pause so you guys get a chance, but I'll give the answers in 5, 4, 3, 2, 1. For A, chlorine is an essential element of life. No, it is not. We only have chunks, so you'll want to, again, remember that. And each letter is an element on its own. There's 6 letters in that chunks, and you want to make sure that you are not confusing that Cl, chlorine, is one of them. B, fatty acids covalently bonded to glucose is considered a polymer. Remember that so far we've only covered 2 kinds. Fatty acids are going with glycerol heads, and glucose is usually bonded to another glucose to create something like what is that one? I think that one's galactose. Anyway, so fatty acids combined to this is not a polymer because we had to have the same monomers in order for that to count. Cellulose is a structural monomer of fatty acids. Um, Again, this is a small thing. I would not test you on examples that we're giving. Personally, I'm not going to ask you to memorize this, but if you go back in the notes, cellulose is not a structural monomer. It is considered a polymer. D, primary function of fat is to provide quick bursts of energy. That's carbohydrates. Fats are long-term energy, generally speaking. Or E, hydrolysis reactions involve inserting water. Remember, we had condensation. Water gets removed out, right? In hydrolysis, you're using water to cut or to break down. So that's what hydrolysis literally means. So you're putting the water in, so it's inserting the water, and then it breaks the bond. So yes, this is true. For F, dehydration reaction can be an example of an anabolic reaction. At least according to this slide deck, you have not yet learned what an anabolic reaction is. So you shouldn't worry about this one for now. But if you were to be asked this, which I'm not going to, not at least according to this slide, if we cover anabolic reactions in a later slide deck, you will be asked this. For now, you have no way to know this, but anabolic generally means you are adding smaller pieces into becoming a bigger whole. So dehydration, which is what we're calling condensation, right? It's the opposite. It's exactly that. You're taking water out and making bigger things, monomers into polymers. For G, ester linkage is a bond connecting glycerol and fatty acids. This one is true. That's the definition of that. It's very straightforward. I would say it's a more difficult version of a tier 1 question because I would usually just ask you, hey, what's the bond for fatty acids or for lipids, right? Not fatty acids. But this is literally the definition. For H, lactose is an example of a disaccharide since it's made from 2 separate monomers. The answer is true, but I mean, you would have to know that the statement itself is true, and you would need to know what a disaccharide is. We didn't cover any of that yet. Disaccharide means literally just 2, so it's not a polymer in the like the poly sense. It's just literally 2 sugars. When you study carbohydrates, they will differentiate. Sugars come in either monomers, like glucose, or disaccharides. So there's monosaccharide, then there's disaccharides, which are very common. Fructose is the sugary sweetness you just taste in fruits. I believe it's made of glucose and I want to say I forget the other base ones. There's so many other base level sugars, but glucose is definitely one of them. And then you have lactose is yet another one, which is combination of some sugars, but it's a disaccharide, and we do consider them polysaccharides because there's more than one, but there are larger chains like starch, which are definitely poly because that's in the hundreds, if not thousands, versus over here you have lactose, which is just 2. All right, so our third candidate, third party member for the biomolecules is proteins. You have the monomer, polymer, and the bond. The monomer is the amino acids, the polymer is the polypeptide, and the bond is known as a peptide bond. This one's fairly straightforward because you just have to memorize the word peptide, and you already got 2 out of 3. However, amino acids is going to be something you really have to memorize. I feel like, at least in my experience, a lot of people do not associate proteins with amino acids. You just have to force that memory into your head. I don't think it's an intuitive term that usually goes too well together. You do it enough, eventually, like, well, of course, amino acids are proteins. But in the meantime, since you don't know that, you're going to have to force that into your brain and say amino acids become proteins. Amino acids become proteins. And the word peptide basically is so associated with proteins, you should instantly think proteins. So, there isn't much to discuss here. We're going to talk about proteins in a lot more detail. And in the future, especially something like OChem, you may have to memorize not just each of the 20 amino acids. They might actually have to memorize their R groups, the things in this case, it's not the things in pink. The pink and blue are the heads and tails of the amino acids. It's the thing that's on the top left that are called R groups. But anyway, if you combine the tail and heads linking together, you'll create polypeptides. And again, in order to create these, you're getting rid of water. What is that called? Dehydration and or condensation. As a quick note, I probably should have brought this up in the earlier slide, but it confuses a lot of people when you say dehydration because you're thinking from a larger perspective. This is where a lot of times people will say you have to study and understand what systems are. And I think if Mr. Q brought up the three businessmen missing dollar riddle, it's exactly the same kind of concept. You're going to confuse yourself if you think dehydration means, well, we're getting rid of water. It's true, but you're getting rid of water from this larger molecule. There used to be water inside of them, but now you're removing it out of their system. In our world, we get to see more droplets of water coming out, and they're missing it. So from their perspective, it's a dehydration. So if any of you are getting confused by that, just understand it's from the perspective change, and suddenly it'll make sense why it's called dehydration. All right, so proteins are, if you will, it's like the Industrial Revolution happened. Okay, I don't like talking too much about evolutionary biology because I feel like it's one of the most misused and abused biological branches. I have so much disdain for anyone that loves to go around prattling about evolutionary biology unless I feel like they're doing some very solid science. Generally speaking, you'll hear me scoff when you bring up this evolutionary biology because I've heard just way too many charlatans try to pretend they understand what it's talking about and misusing it like crazy. But if we were to speak evolutionary biology, if we're talking about as far as we can guess, in the evolution of things, proteins became such an important piece. Once cells knew how to start using them, suddenly you were able to create so much more than just basic levels of like cell. You can actually create a lot more things. This is called, as Mr. Vo puts it, building blocks for life. There are just too many functions to even begin. When you guys study a lot more of these, you'll begin to see why proteins are so important and why we say, oh, muscles are made up of proteins. And, there's a ton of things that proteins are so important. Remember ribosomes when we talked about those from cell organelles? They're basically pure proteins. And it's incredibly fascinating to look at these things and say, hey, what are proteins able to do? There's a ton. There's a ton of reasons that they exist, and it just depends on how you build them. All right, with the next one is nucleic acid. And if you over here, Mr. Vo wrote DNA and RNA, for the most part, there's no other reason for us to study nucleic acids. There's other things that they do, but for all intents and purposes, for not just us, but also AP Bio and most biology, when we talk about nucleic acids, we're just talking about DNA and RNA. So in this case, their monomer is known as a nucleotide. If you think nucleus, right, nucleic acid, nucleic, nucleic acid. The polymer is known as nucleic acid. That's it. That's partially why Mr. Vo wanted to have DNA and RNA on top, is because if I say a nucleic acid's polymer is a nucleic acid, it can get a little confusing. But that's how you're going to have to memorize it. Nucleotide is a single one, that's a monomer. Polymer is a whole acid. Now the bond is called a phosphodiester bond. Again, We're asking you to memorize that. The earlier you guys know these terms, them associate them with the right things, the less you'll get confused. Because later you'll actually be expected to know this. Right now it's just it's extra information, but we're forcing you guys to memorize them. And finally, the function of nucleic acid is to store genetic information. We're going to talk a lot more about these when we start getting into DNA and reproductions of cell, but for the most part, just remember that they're storing genetic information. Now, there is a secondary job, and it is known as temporary energy carriers. ATP, NADH, and FADH2, you will hear a ton of. If Mr. Vo decides not to go too deep into this, this is probably as far as you'll hear it. But in AP Bio, for sure, you're going to memorize, I think, what, three, four, five long chains of like reaction after reaction after reaction to explain how the cells use ATP, use glucose, and maximize the amount of energy they can get out of it. You will need to know NADH and FADH2 and also ATP. In fact, some classes are insane. They'll make you memorize the exact shape of ATP, like it's shown down here. You'll notice that there's an ester bond in there, and they'll ask you all kinds of crazy molecular questions. For all intents and purposes, we're not going there. You just have to memorize it as temporary energy carrier. And that does it for our biomolecules.